Welcome to the first part where I want to explain the objectives and motivation of the study to you. Even though all transitions are important, the transition from fruit to dispersed seeds, this transition is particularly critical at different levels for the plant dynamics. For example, at a large population scale, seed dispersal determines the likelihood of colonization of different subpopulations. At an intermediate population scale, seed dispersal determines the probability of colonization of vacant habitats. More importantly, at a very local scale, seed dispersal determines the initial template in which all post-dispersal processes, such as germination, seedling growth, and survival, predation, etc., will be manifested. For example, seed dispersal determines whether the spatial pattern of recruits, that is, seedlings, is clustered or scattered. This in turn determines the level of intraspecific competition between seedlings of the same species, and also the likelihood of heterospecific competition between seedlings of different species. On the other hand, the spatial pattern of seed dispersal is also likely to affect the likelihood of interaction between the plant recruits and the animal partners. For example, in general, we would expect that generalized herbivores would prey more intensely upon aggregated than upon scattered seedlings. For instance, deer would tend to predate on seedlings that are aggregated than on the other seedlings, because they are easy to find. However, some trophic specialists, like snails, can predate on particular species of seedlings, regardless whether they are dispersed or scattered. Also, there are some habitat specialists that can focus on seedlings that are in a particular habitat, like the rabbits. Some rabbits, they can focus on seedlings that are in a particular habitat, regardless of the species and regardless of the spatial distribution. So, because the spatial pattern of seed dispersal are so important for plant performance, we investigate three mechanisms of seed aggregation. The first mechanism, we will investigate the three potential hierarchical mechanisms of seed aggregation, that is, and they will be to answer the following questions. Do frugivores spatially aggregate the feces, and if so, at what spatial scale? The second question, do frugivores deliver feces containing seeds aggregate within the overall pattern of feces? And if so, at what spatial scale? And finally, is there any association among the number of seeds of different pairs of species, and if so, at what spatial scale? So, seed dispersal is certainly a critical transition in plant dynamic at different levels, but as I mentioned already, seed dispersal by animal is also a very complex process involving many potential dispersers and many contrasting plant species. Therefore, we need to use powerful tools, analytical tools, that allow us to describe in detail the spatial pattern of such complex processes. Fortunately, spatial point pattern analysis deals with the statistical analysis of map point patterns, which comprise the coordinates and additional features, such as species, size, fitness components, etc., of ecological objects. These ecological objects could be fecal samples comprising seeds. This sort of analysis relies on several assumptions, such as, for example, that all points in an area or a representative sample of them are considered. The objective of this study is to illustrate how spatial point pattern analysis can help us to reveal this hidden information in complex pattern of seed dispersal by animals. So another objective, as I said, objective of this study is to illustrate the usefulness of spatial point pattern analysis to reveal such a hidden information. Therefore, in the next videos, I'm going to introduce the study size and the field methods. In case you want to deepen your knowledge of the statistical background, please watch the corresponding videos.